I'd like to thank the opportunity to come to Toronto for the first time. Considering I'm half Canadian on my mother's side, since she was born at St. Michael's, I think it's kind of an opportunity to explore. And so I'm going to try to take something that none of you were probably used to thinking about, which is tumor metabolism. And one of the things that's interesting about tumor metabolism conceptually is it's a bunch of enzymes. Enzymes are inhibitable. Tumor metabolism and standard metabolism are very different things. And this really does lead to the next level of therapies. And this is where all the new drug targets are coming out of. So because Robin's here, top that. I just had to update it, actually.
I just changed my slides.
Yeah. So, it was a very sad ASCO in the plenary last year. I mean, I think that my entire career had started with Olertumab, but I was the first person that actually treated the first person on the phase 1 of Olertumab, and my entire, trialist career kind of collapsed when Olertumab was basically shown to be an error. But I could not have paid the discussant to say this any more than I did, because I think that this is something we should be thinking about in terms of where we're going to go with cardiac sarcomas. But what he said, and I thought this was very intuitive, is we need a biomarker-driven therapy with a mechanism for sarcoma if we're going to go after large groups of tumors. Because, I think one of the things that you shouldn't be doing is throwing intimal sarcomas in with angiosarcomas and leiomyosarcomas. These are so molecularly different. And, we have to respect the fact that the parallel word to sarcoma is not breast cancer, it's carcinoma. We are now, according to CTOS 2019, treating 175 diseases with doxorubicin and ifosfamide and expecting to see differential results. And so, we started teasing a lot of this apart. And what you'll see is we're going to end with a biomarker-driven therapy for about 90% of sarcoma that's mechanism-driven. And so we're going to build up to this, and I'm going to hope you can handle or stay with me. So I'm going to try to keep this simple. So mTOR is not metabolism. mTOR is a signaling tyrosine kinase. It is not metabolism. What I'm talking about when I actually say tumor metabolism is carbon. Carbon that comes from glucose, carbon that comes from glutamine, carbon that comes from lipids. Tumor metabolism comes from the concept that the purpose of cancer is not to kill you. The purpose of cancer is just to make more. And to make more, you need to make more RNA, more DNA, more protein, more lipids, more amino acids, and all of that comes from the carbon sources that are in glucose. And if you don't want to use glucose, you can just break down your amino acids. A lot of things work on glutamine. So when your PET goes down, you may have just adapted your tumor to an oxidative phenotype. And so these are things where I think that we have, as medical oncologists, a need for a deeper knowledge of what we're doing. And I think groups like this are the ones that are going to take something that's really challenging, like cardiac angiosarcoma, and do something about it. So conceptually, I like PowerPoint, you're going to learn. Enzymes and metabolic pathways, you think of them like gears of a watch. So what's interesting is you can go from here to here and over to here, right? And so you're able to get from this side to this side. And then if I look at you funny and try to inhibit things, well, now I can go this way and then go this way and this way. Enzymes go in both directions, and there's so many reiterative pathways that this is actually hard to develop single-agent drugs in. I mean, if you look at glutaminase inhibitors, if you look at arginine deaminase, if you look at a lot of the metabolic therapies, unless you are drugging a driver like the IDH inhibitors, you really have a hard time really getting single-agent activity, which is a challenge to drug development as we know it, because these things all have to be developed in combination. So in cancer, though, it's kind of fascinating. You can go around and around and around this pathway because, well, you need it. But sometimes we shut things off because if you're going to make biomass, you may not want to have a sink. And so cancer shuts all sorts of things off, brings back all sorts of embryonic development genes. And these, you realize that blastocysts live on glutamine prior to implantation, as opposed to glucose? we bring back blastocyst biology. And so what this really means is that if we can understand tumor metabolism, we're going to understand potentially a whole new avenue for drug targets. And so this is kind of a normal PET scan. When glucose goes into a cell, it's going to go through glycolysis, and I'm going to try to keep this simple. Don't worry about the words. And pyruvate usually gets made down to lactate. What cells are not doing is making nucleic acids, unless, it's a hair cell or a stomach cell, or a colonic cell. We're not dividing. We're not making a lot of the Syrian biosynthetic pathway metabolites. We're not making phospholipids. We're not making a lot of amino acids in terms of what we're doing from glucose, because we don't have to make more. We're at homeostasis, right? We are eating. We have most of the nutrients we need. We're not going to double in size unless you live in Missouri, where they double in size quite readily. But I think most of that's brought to you by McDonald's. Not a sponsor for today. If you get into cancer metabolism, though, things change. The first thing that happens in general is that you express that blastocyst pathway and you block this, right? You're no longer using glucose for energy. You're all of a sudden going to start making nucleic acids, phospholipids, and amino acids. And what these are the pathways that branch off glycolysis. It doesn't matter what the enzymes are at this point, but what you see is what you're doing, right? And, there's all sorts of fancy rheostats that sense where the tumors are. This is elegant biology, and it's the biology of a blastocyst, which is the biology of cell division, how one cell becomes somebody who sits here, right? There's a lot that we as sarcoma doctors can learn from our leukemia colleagues, right? Metabolism has been used to treat cancer for an incredibly long time. So L-asparaginase, which is going to make a comeback for sarcoma soon, seriously. There's this really, really neat company in France that's found a way of giving asparaginase that I can talk about later. So asparagine is this dispensable amino acid unless you're missing an enzyme to make it, right? And so you have essential amino acids, non-essential amino acids, and conditionally essential amino acids. And so both of those are asparagine and arginine in terms of what we can modulate. So asparagine is dispensable as long as you can synthesize it. ALL lacks asparagine synthase, which makes all the ALL cells dependent on extracellular asparagine. This is why you give asparaginase. And so if you deplete extracellular asparaginase, this leads to protein synthesis inhibition, which is a fancy word for starvation. That leads to autophagy and apoptosis, right? So all of a sudden your blood cells can still make asparagine and process it, but the cancer cells can't, and then you die. This is good, right? We want to target cancer, but not the rest of the body. But we know all the problems that come with asparaginase. And so I've been working now for way too long on the world's worst named enzyme. But we're going to keep going. And so can we find the ALL parallel for sarcoma? And we're going to need a biomarker, just as was said at ASCO. At least a pair of drugs. And right now I'm at 3, going on 5. And clinical trials. And I think one of the things, whether they work or not, if you are not taking what you're discovering in clinic and putting it into trial, you're doing not only the patients a disservice, but you're also not actually proving what you're doing is right. And hence why we're all walking around asking questions about propranolol and whether or not it works, because it's not been done in a prospective randomized trial. So there's a lot of biomarkers that are actually exciting. And so the bottom 3 are mine. The top 2 aren't. But there are papers circulating that I don't know if they're out now on IDH inhibitors in chondrosarcoma. There is a basic science paper showing about 50% of dedifferentiated chondrosarcoma has mutations in IDH 1 or 2. And I know that there was an Agios trial that may or may not have been reported looking at that drug. There is a different company that I'm involved in that actually is looking at IDH mutant chondrosarcomas and other cancers. These drugs are well tolerated. You're drugging a driver of a pathway you're not addicted to, right? This is kind of neat. And so that's out there, that's evolving. Last year at Keystone, Tumor Metabolism, the biology of SDH deficiencies and what actually happens within the TCA cycle was cracked by a colleague of mine in Glasgow named Gottlieb. And it's an exciting pathway that might be druggable. There's all sorts of pathways we don't know about in metabolism. But this is, paragangliomas, wild-type GIST. This may open up avenues for treatment. Arginosuccinate synthetase 1 deficiencies is actually the topic of what we will talk about today. There is no ASS1, there is no ASS2, and I'm stuck with ASS1. We're going to talk about me deficiencies in synovial sarcoma, which is a paper that we'll come out with soon. That's completely druggable in terms of synovial sarcoma. And I'm going to take everybody back to phosphoglycerate dehydrogenase deficiencies in osteosarcoma and Ewing soon. All of this are metabolic targets within different sarcomas. If you respect the fact it's 175 diseases and start to take it apart, what you realize is only one biomarker here seems to translate across all of them. And so about a million years ago, when I was first on faculty, Brian Rubin had done this brilliant experiment where he looked at 701 sarcoma specimens in a TMA of 45 different histologies. And what he found is that 88.3% of the time, if you score things 0, +1, +2, +3, and only look at the things that scored 0, 88% of the time there was no functional expression of arginosuccinate synthetase 1. And what this is the rate-limiting enzyme in the urea cycle to make the amino acid arginine. And this suggests that the most common defect in sarcoma is the silencing of ASS1. P53 happens around 50% of the time, give or take the study. This is almost 90%, right? And it's irregardless of histology with some interesting things to point out. First, solitary fibrous tumors, which are solitary and non-metastatic, don't seem to silence it, right? That's interesting. Some of the myxoid liposarcomas don't seem to silence it. And synovial sarcoma has an interesting biology. But in general, as you go through things, this is a recurrent mutation in most high-grade sarcoma. It's not mutation, silencing that occurs in most sarcomas. It's never mutated. You can't be born with a mutation in ASS1. You get a disease called citrullinemia. You die of high levels of ammonia, which is this is an enzyme you just silence. And so basically, this is a gene that's on chromosome 9. And there's nothing near it that I think is standing out to me that's interesting. And so just to review the urea cycle, because I'm sure everybody has seen this since medical school. So if you take bicarbonate and ammonia, you go through CPSI and you make carbamyl phosphate. You go through a gene called OTC and you make citrulline. Citrulline you actually care about on this, okay? Citrulline goes to arginosuccinate synthetase 1 to make argininosuccinate, and this is why. Aspartate. And we'll get into why you care about aspartate. Aspartate is how you make nucleotides, right? You go through lyase, you break out fumarate. Fumarate goes to the TCA cycle, and arginine can then be either used to make nitric oxide, which will make all the cardiologists in the room happy, or ornithine. And so that's the urea cycle, right? You're taking ammonia and making nitric oxide, you're making arginine, you're excreting urea. Cancer puts a block here because it can get all the extracellular arginine it needs for nitric oxide production from the bloodstream. But by not using aspartate in the urea cycle, it is freed to make nucleotides. And if you're only concerned about dividing, you don't care about making nitric oxide as much as you care about making nucleotides. And so there are a number of drugs out there that'll destroy extracellular arginine. And so targeting the inside of a cell with something like arginase doesn't work for our tumors because they're not making their own arginine, so what's in the cell isn't going to be broken down. So there's 3 ways of going about this. You can use arginine decarboxylase, or arginine deaminase. And so you can send your metabolites to different parts of the urea cycle. So arginase catabolizes arginine to ornithine and has a low affinity for arginine and works at a very high pH. And so if you have a septic patient that you want to treat their cancer, this may be an option. Arginine deaminase actually sends you from arginine to citrulline right before the block. So you get a buildup of metabolites before the block that can't be used. And this has been in phase 2 and 3 trials for about a decade. Most of the phase 3 trials failed because they tried to develop it as a single agent. And so basically what arginine deaminase does is take this nitrogen and make it an oxygen, which is the difference between arginine and citrulline. It's a single little nitrogen to oxygen. And what you find is this is a shot you give your patients once a week. It has 2 side effects. You can get a rash from the injection site, and it can cause gout. I have yet to see gout happen, but I have seen the rash. But it's well tolerated. The most frustrating part about an enzyme in terms of clinical practice is if you just give the shot, the patient has to wait for the ice cube that is in to melt because you can't heat an enzyme. So they sit around clinic waiting for 2 hours for ice to melt, and so you have to temper how you tell your patients they're going to get this. And so we have gone off, and this will be the most complicated part of the lecture. And so I'm going to try to keep it simple, but I apologize ahead of time because metabolism, when you get hardcore, is hardcore. So I have a tendency to work in systems biology because it's very hard to publish a sarcoma paper. But if you publish the fact that arginine deaminase works in sarcoma, pancreatic cancer, small cell lung cancer, and all does the same thing, then the journals care. And so what you're going to see is SKMEL2 is actually a melanoma cell line. SKLMS1 is a leiomyosarcoma cell line, and SKUT1 is a uterine tumor. What they have in common is they don't express ASS1. And so this is a fancy little assay that shows that when you treat cells that don't express ASS1 with arginine starvation, you induce starvation. Proof of principle, right? And so you get these autophagosomes that basically start destroying the intracellular arginine to repurpose it for things that keep you alive. What this shows you is that when you starve cells of arginine, they fall out of cell cycle. And so if you look here in orange, that's the G1 peak, this is S, this is G2. What you find is you kind of get this loss of cells in G2. They just stop dividing where they are because they can't. They're missing the metabolites to keep going. They fall out, they enter this starvation response to try to survive. And one of the things that you want to do is survive. You don't necessarily need to continue dividing. But then you find a way to escape, right? And the easiest way to escape from your arginine starvation is just to re-express ASS1. That actually turns out to be the most useful thing that we've ever discovered. So we then went after all sorts of concepts of synthetic lethality, and we've actually found 3 and now 4 answers, one of which isn't part of this lecture because my postdoc would kill me. So the first thing is I can conditionally make cells dependent on glutamine. We're going to show you the data. And when things are conditionally dependent on glutamine, glutaminase inhibitors become cytal. They're not just static, they're cytal. We can actually make cells dependent on the Syrian folate cycle, which is a pathway that I just love. I mean, people have been studying Syrian folate, high-dose methotrexate. They've been studying this pathway for generations, but we've actually made it conditional. Then inhibitors kill. And finally, gemcitabine is a metabolic therapy. And I need you to start realizing gemcitabine is not chemotherapy, it's a metabolic therapy, because when you inhibit the ability to make DNA, because you've inhibited the ability to produce the nucleotides, gemcitabine actually functions as a metabolic therapy. So let's scare you. So what you're going to see is that we do a lot of metabolomics, and what you'll see in blue are cells at baseline. Red are cells that are short-term arginine starved, and green are long-term arginine starved. And so there's a metabolic adaptation. And all you care about are the things that happen in these 3 circles. The first is glutamine. So the longer you treat a cell in arginine starvation, the less glutamine you find. And the most conceptually hard part of metabolism is to realize that sometimes down is up. So if you don't see a metabolite, you're either not making it or you're really using it, right? And so I'm going to show you the data that shows that we actually are going to switch these tumors from glycolysis to complete glutamine dependency. Their PET goes down and their tumors grow. Glutamine PET, on the other hand, goes up. I like PET scans that go up with response because that's harder to futz with, right? you'll see this peak in Syrian. All of a sudden, we drove all the glucose through the Syrian pathway. It's kind of an interesting observation, and I'll show you that data. And finally, you'll see that there's all sorts of changes in lactic acid biology, which really show that you change what you do with glucose. And so let me show you the data for why we're trying to push the NCI to help us do a trial with ADI and a glutaminase inhibitor. So Calithera has a glutaminase inhibitor, and it's actually quite a good one. And it's something that I'm very protective of. It's something I truly believe in. And it's something that I worry doesn't work well as a single agent, because the easiest way to get around glutamine is just to switch back to glucose. There's too many ways to solve a metabolic question. But CB839 is this wonderful glutaminase inhibitor. And so let me show you what glutamine does. If I just pop this cell up, you'd be scared. But as glutamine enters a cell, there's a wonderful transporter that brings it in. Glutamine is then either goes through a cytoplasmic glutaminase to glutamate, or it goes into the mitochondria and goes through glutaminase to glutamate. This is the only time you can stop it. Past here, what you see is there's a lot of ways to make alpha-ketoglutarate. And once you get here, this gets complicated because you have to start using 5 or 6 drugs to stop it. So this is why glutaminase is such an attractive drug target, right? Once you make alpha-ketoglutarate, it goes into the TCA cycle. The TCA cycle rolls around, and all of a sudden you start making biomass from glutamine. So you can shoot out into lipid synthesis. You can go and shoot out into both aspartate and pyruvate. So this is energy metabolism. This is also lipid metabolism. It's also nucleotide metabolism. So you could actually start using glutamine to make everything you actually would make from glucose, right? But it's got this wonderful, wonderful point of inhibition. And so remember I told you that glutamine went down. And what we did is all of a sudden we were able to find a lot of glutamate, and we started tracing it around to figure out where it went. And so you can take these heavy labeled carbons. It's carbon 13. It's not radioactive, but it's just heavy labeled. And you can then do mass spec and say, where do heavy labeled glutamine carbons end up? And when you treat with ADI, one of the things that happens, and this is wonderful for radiobiology, is you say, well, where does glutamine go? Glutamine goes to glutamate. You'll see that it's still down early. You can say, well, maybe the glutamate's all going into glutathione. And the first thing you find is you lose your glutathione pool. This is useful for radiotherapy, right? So everybody's always wanted to get rid of glutathione. It's your antioxidant system that helps protect you from dying. Well, this depletes this. But this also told me that when we rewired this cell for pro-survival, we didn't go this way. If you look then in the TCA cycle, you're going to find that when you treat with arginine deaminase, malate and citrate levels are really low. But what I'm going to propose to you is that actually they're down because they're up. Because what you do is then you trace them out of the TCA cycle and you look at aspartate and asparagine, and what you find is you can finally find the accumulation of where all the metabolites end at asparagine. And it was at 24 hours when you trace all these things around where all the carbons ended up. And asparagine this is all nucleotide biology. It's all nitrogen sinks. It's all the things you want to make more. And so one of the more simple things you can think about is these cell lines, they all started off as glycolytic, and in this corner you're looking at extracellular acidification. It's like how much lactate are they making? How much are they using glucose? And as we treat the arginine starvation state, you'll see that all 3 of these cell lines are making less lactate, right? Glucose is doing something else. But then if you look at mitochondrial function, just by their oxygen consumption, what you see is that this starts going up. And this says, okay, we've turned on the mitochondria. We're dependent on the mitochondria. It's all this glutamine biology going through. And we said, well, that's interesting. We turned off the Warburg effect. Glucose being used to make biomass. And we made everything addicted to the mitochondria. Well, remember I showed you the entrance into the mitochondria was through glutaminase. So then we did this simple little experiment. It wasn't so simple at the time, where we watched cells grow. And just as expected, when you treat cells that aren't addicted to glutamine with a glutaminase inhibitor, nothing happens. This is every cell in your body. Isn't this kind of cool? If you treat cells with arginine deaminase, they stop growing for a while. If you add a glutaminase inhibitor, though, to cells that are in the arginine starvation state, they start dying. And so all of a sudden we're able to use a biomarker-directed, first drug works against the biomarker, second drug works because the first drug works therapy. It's sort of the R-CHOP idea, right? Except I'm doing it against a biomarker. And notice arginine deaminase doesn't do anything to normal cells. Glutaminase doesn't do anything to normal cells. This is a tumor-directed metabolic therapy, right? This is something where you now go after the tumors specifically. And so at the time this was done, there wasn't access to Calithera's compound because it was under development. And what you found is that if we knock down glutaminase, these, or just short hairpin to GFP, the tumors grew normally in a mouse. If you treat it with arginine deaminase, you got this slow escape over 30 days. If you didn't have the ability to switch to glutamine biology by the knockdown of glutaminase, the tumor shrank in vivo by about 50%. So you're actually able to do this in a mouse, which means that we should really try doing this in people. I'm going to kind of cursorily tell you about why the Syrian folate pathway is important. So we then not only heavy labeled glutamine, we heavy labeled glucose and asked where it went. And what we found is it wasn't going through the pentose phosphate pathway. It wasn't helping make ATP. But we got this huge spike in glucose going into serine and then into glycine. And this is a single carbon that goes down to the mitochondria. You found a loss of lactate, you found a loss of citrate, so you weren't taking glucose and putting it into the TCA cycle. You weren't making lactate. You're actually sending it all through serine. And I said, well, what's going on? That's weird. So we then looked at the rate-limiting enzyme and entrance enzyme into the serine folate pathway, which is phosphoglycerate dehydrogenase. And these are untreated cells here and here. And this is what happens in the arginine starvation state. You clearly upregulate the rate-limiting enzyme and drive all your glucose this way. It's also sending the carbons that you want into the mitochondria for biomass. And so my student made a whole bunch of conceptually cute little PowerPoints, and I'm going to try to keep this simple in a very complicated way. So when glucose goes through glycolysis down to pyruvate and lactate, you can go through pH, GDH, but you can actually inhibit this with CBR 5884. And what you see is just like with the glutaminase inhibitor, the pH ADI stops things from growing, and this is actually death. CBR doesn't actually do much by itself, and the combination actually starts to induce significant cell death. So we went down a little further into the folate cycle with a drug that we actually know how to use, which is methotrexate. And look, all of a sudden they make methotrexate kill much better, right? We have a targeted therapy. Methotrexate we don't think of as targeted, but it's actually very targeted, right? It's one of the better targeted drugs we have. It's just really toxic. So if you think about what's going on, and this is kind of a big picture view, glucose goes in through a cell, and you either make lactate or acetyl-CoA. And so if you put in this PKM2 block, you then realize that's cancer. If you put in the arginine starvation state, I can actually remove this block. But the cell doesn't like it, so it actually phosphorylates pH-GDH and actually gets rid of LDH. This is why everything's going through serine, because the cell found a pro-survival mechanism. If you can make things dependent on a pro-survival mechanism, you can kill them. And that's all through pH-GDH, which is something you're all going to be learning about soon. And so what's interesting is I told you we'll go from serine to glycine, glycine down to pyrimidines. And this is where we're going to end the talk with the last section, which is something that is now in trial. And so we found out that docetaxel, I'm going to show you today, is not a mitotic inhibitor. Docetaxel is a MYC inducer. Don't know why. Gemcitabine is a metabolic therapy, and arginine deaminase is a shot. And so we have a clinical trial open right now at WashU, Stanford, Santa Monica, and soon to be Columbia, and it's the only crowdsourced trial I'm ever aware of. Everybody's paying to be involved. I'm not paying anyone. This was started as an institutional IIT that people were so excited for that the rich friends of mine at Stanford and in Santa Monica said, well, I can make money off of billing for GemTax, so we'll just throw in the trial for free. It was awesome. I mean, I can't thank Kristen and Sant enough. And so there's a recent paper in Clinical Cancer Research called Arginine Starvation and Docetaxel Induced c-MYC-driven HENT surface expression, which is the transporter for gemcitabine. It overcomes transporter-level resistance. And this is why focusing on something for a decade matters, because I've been pushing this for a decade. So one of the early pieces of data that didn't make our cell reports paper, but that we did know about, is that SKLMS in a cell culture is the classic gemcitabine-resistant cell line. It doesn't care. You can grow it in like 1 molar gemcitabine. It'll grow through. In the arginine starvation state, once you grow through the arginine starvation state and adapt, you become sensitive to gemcitabine. And so we pursued this. And so I need to probably educate everybody, since I'm sure everybody has this memorized. Arginine goes in through CAT1, and so this is an important transporter, and this gets upregulated if you're in the arginine starvation state. Gemcitabine goes in through HENT1. It then must be phosphorylated by DCK. DCK, once you phosphorylate gemcitabine, it's trapped in a cell. You go through multiple phosphorylations, and then a cell has to decide whether it wants to use gemcitabine or cytidine. And this is controlled by the expression of RRM2. If you don't have RRM2, you preferentially use gemcitabine. And I'm going to show you a perfect storm, which is really good for our patients. And so if you're ASS1 positive, this is your biology. You have to rely on natural transporter expression. If you're deficient, we're going to go here. And I'm going to build this out for you. And what you're going to see is that once you don't have arginine, docetaxel will drive CMEC to the nucleus. This will drive HENT to the surface. I'm going to get gemcitabine in the cell. I've got to upregulate DCK to, I'm going to upregulate DCK to phosphorylate it. I'm going to get rid of RRM2, and then I'm going to kill the cells. And I'm going to show you, probably not in this presentation, but we did this paper looking at melanoma, pancreatic cancer, and sarcoma. And so this is ASS1 biology and not sarcoma biology, but the good thing is my loyalty to the sarcoma field is why all these trials are in sarcoma. And so if you first look, docetaxel, if you look at what, in terms of growth inhibition, doesn't do much. I'm actually surprised it works in angiosarcoma. It works quite well in angiosarcoma, but in terms of actually in cell culture, it doesn't do much. And things just grow right through it. But one of the things we noticed early on, we were doing a control experiment, and in the arginine starvation state, if you add docetaxel, we noticed the expression of SLC29A1, which is HENT, went up dramatically. And this was just a control experiment. And then, you love it when your postdoc gives you data and you're like, well, this is cool. That shouldn't have happened, right? So if you actually then take an antibody, and this is a real-time experiment. So if you look at cells that are untreated and you look for the cell surface expression of HENT, everything you're looking for, which should be these little yellow things, it's not yellow. There's no gemcitabine transporters on the surface. In the presence of docetaxel, this real-time assay shows you all of a sudden the cells start to coat themselves in this transporter from gemcitabine. And we thought, well, that's useful, right? So then we said, well, what if you had gemcitabine? And so if you look, traditionally, no matter what your dose of docetaxel is, adding gemcitabine, the cells were resistant. You put them in the arginine starvation state, all of a sudden you started to kill. And the longer you cultured in the arginine starvation state, the more they became sensitive to gemcitabine. This is a conditional-driven synthetic lethality, where it's not the mitotic inhibition of docetaxel, it's this induction of MYC. And so what you see is that if you look over here in the gemcitabine pathway, if we look at DCK, which phosphorylates gemcitabine and traps it, you see that you're actually able to dramatically upregulate its cytoplasmic expression. So this is a fractionation looking at nuclear and cytoplasmic, and this is a cytoplasmic expression where you need it to trap it, right? If you look at what happens to RRM2, which is the choice between cytidine and gemcitabine, you lose it from here. And so it goes down dramatically. And so all of a sudden you're selectively using gemcitabine. And so then if you look at all the combinations of cell death over time, you get minor cell death with gemcitabine and docetaxel at baseline. You get a lot of cell death with the combination. And so we then went and said, well, is this CMYC independent? And what you'll see is if you use a MYC inhibitor, you can block the entire biology. So don't block MYC if you want to use this strategy. This is not the thing to mix it with. But we also then made a FITC gemcitabine analog. If you look at the uptake of this analog, it goes up. The longer you treat with arginine starvation, the more gemcitabine fits the analog you can uptake, right? This is proof that you're actually taking up the analog. And you can block the whole thing with a MYC inhibitor, once again proving don't give MYC inhibitors with ADI. And this is just kind of the pictures that show that. Then we did a synergy experiment. This is something that I've been pushing everybody in the world to start doing. If you actually ask, are gemcitabine and docetaxel synergistic? Actually, most of the dots for them are up here. It's the most antagonistic combination you'll ever see. If you stop a cell from dividing, how are you going to use a chain terminator? As you go into the arginine starvation state, what you see is then you begin to get synthetic lethality, and if you go longer, you push it. So you become you have much more synergy. You want something that's synergistic and not antagonistic, right? So it always begs the question, and I think that's why Anthony Elias doesn't believe in docetaxel. He must have a good feel for this. So then we finally did the animal experiment, and I think this is where we learn a lot. So SKLMS1, if you grow it in a mouse, grows out in about 28 days. And so now I want you all in your head to picture what you see in nature, which is like 14-day growth curves only to about 500 millimeters cubed, okay? And so if I had done that, you would think, oh, look, ADI works great. Docetaxel doesn't do anything in vivo. ADI and docetaxel slightly amplify, but I think one kind of slows you down and they kind of work a little bit. But look, I had a winner here. If you just look at 500, I could get you 3 stars. Or I could give you gemcitabine. Now, this is starting to get meaningful. So if you take something from 28 days to 56, that's a doubling, right? That's something that's probably meaningful. If you added ADI to gemcitabine, what you actually saw is tumor shrinkage a little bit, but then they escape at about the same time. It's interesting in vivo that the winner is ADI, or is gemcitabine and docetaxel, until you throw in ADI. And then all of a sudden, you're now moving much farther this way. And so what you're seeing is that we're able to really start to harness tumor metabolism to really direct MYC biology and transporter biology. And what I thought was really interesting, this paper comes out in Clinical Cancer Research. There's a paper in Cancer Discovery the same week talking about how macrophages throw nucleotides at tumors, which is why they downregulate their transporters, because they get what they need. So taken together, you go from a model where cells kind of take the arginine they need in to survive, and you're kind of hit and miss with the gemcitabine pathway, to one where arginine gets destroyed, and in that condition, docetaxel drives your MYC to put this transporter up here as a pro-survival mechanism that you can then use to kill. So in conclusion, there's a lot of work to do in metabolism, and I think with the eye that we're going to translate it, it's really kind of a neat area to be in for development, because I am not an immunotherapist. Sarcomas, because of their frequent ASS1 deficiency, are primed for this metabolic-based therapy. Presently, we're trying to build on the idea of a biomarker-driven metabolic mechanistic therapy. And clinical trials need to happen, and I'm hoping that this group will start launching them. And so thank you all. Hopefully I didn't run over too much, but there's a lot of people involved. The early work was done by Jeff. Bethany did all the transporter biology, and we've been supported by a large group of people over time. And we've had lots of funders, including the SFA, SARC, and the NCI. And so from that, I thank you.